let us move on. So our next speaker is Dr. Lakshmi Mundkur from the Biological Research uh, uh, Division of the uh, Sami-Sabinsa Group Limited in Bangalore. Dr. Lakshmi's team, she's the vice president over there in the biological research team, and her team focuses on the natural products, probiotics, gut health, mucosal immunology, metabolic diseases, and clinical evaluation of various natural products. So, Dr. Lakshmi, after post-graduation in biotechnology and PhD in immunology from the Madurai's Kamra University, she started her career as a research scientist in lupin chemicals and then as assistant professor at the Institute of Science, Aurangabad, Maharashtra. And then she worked as scientist and principal investigator in the Oak Research Center, focusing on new generation proquinolones, oxazolidines, and their molecular mechanism of action. Then she also worked at Novartis Healthcare Hyderabad, where she focused on understanding the clinical trials across the globe and analysis of data for scientific publications. And later on, she worked as the head of molecular immunology at the Thrombosis Research Center in Bangalore, where she undertook basic research on cardiovascular diseases, and she published numerous high quality work from the from her stay during the Thrombosis Research Center. And I happened to meet her there at the time when she was there. And so Dr. Lakshmi would share with her uh, expertise in the drug discovery and pharmaceutical biotechnology, particularly for the natural products today. So we welcome Dr. Lakshmi and request her to present our work on this area.
Thank you, Dr. Nitish. It was, yeah, I remember meeting you at TRI when you had come as an external examiner. Yeah.
Yeah, it was nice
So,
to see you. Yeah,
yeah, true. Long time since that happened.
yeah,
Yeah, so I'll try to finish my talk as within the time that is given to me.
sure.
So I can you see my
Yes, yes,
yeah. So I'll focus today on the drug discovery and pharmaceutical biotechnology, a little bit on the natural products. Can you see now?
yes, it is coming. Yeah, yeah,
Okay. Yeah. So I'll move on.
yeah,
So
it
this
moves.
is
Yeah,
the
yeah.
overview of the today's talk. So I'll just talk a little bit about the drug discovery, the process, and then the use of biotechnology in development and natural products, the challenges with natural products, and a few examples and how we have used these techniques to develop new drugs. So these are all very, like, this is a very vast topic, and there are several reviews. Several people have written a lot of reviews on these topics. So, but to give you a basic outline, the phases of drug discovery involve the first aspect, which is discovery and development, which moves on to preclinical research, where you evaluate whether your target or your product is working in animal models. Then it moves on to clinical development, and then it goes to FDA for review, and then there is post market research, where the drug is further evaluated in a vast population. Now, the step one is the discovery and development, which is the most important aspect of drug development. It starts with the understanding of what drives the disease. What disease are you interested in, and why does the disease develop? And then the new insights into the process of the disease. This allows the researchers to design a product to stop or reverse the effect of the disease. So classical pharmacology to investigate, like chemical libraries, small molecules, natural products, or synthetic molecules, hybrid molecules. These are the different molecules which are used as a therapeutic agent. Now, it started way back in 1900 when the drug discovery process started. So it was basically medical chemistry and the phenotypic screens. What I mean by phenotypic screen is like whether the molecule that you're talking about is bringing up the final effect or not. So that was the main aspect of screening the drugs. Now, slowly, as different biotechnological techniques developed, so it went on to receptor biology and then recombinant technology, combinatorial chemistry, and high throughput screening methods. So the process of drug discovery became more target oriented. And now, like in the recent 30 years or so, we are moving back again to the phenotypic screening. Now, this was the early stage which I was talking about, the selection of the disease. So the first process, this is a target-based screening where you first select the disease that you're interested in. For example, you're talking about diabetes. Now, what is the target that you're selecting for diabetes? There are so many processes which are involved in a disease. It's not that there is only one target which can be used as a target, a protein which can be inhibited and the disease will be cured. It does never happens like that. So one target for diabetes, are you talking about the DPP4 or the dipeptidyl peptidase inhibitors, or you could be talking about insulin secretion, or you could be talking about the reuptake of glucose. So these are all different targets which uh, you can develop a drug for. So if I take the DPP4, then if I if I'm able to get an inhibitor for DPP4, then I will probably prevent the degradation of GLP, and then have the glucose levels will be reduced in the peripheral system. So that is how the drug development started. So the target ID, and then you develop an assay for the enzyme, and then you have high throughput screening methods, and then you optimize the lead, preclinical, clinical, and then FDA approval. Yeah. So am I with you?
Yes, yes,
Yeah.
yes.
So then came the phenotypic screening, where one of the where the disease is selected. Now, any product, if the first level of screening is going to be either in the animal model or in the cell-based system, but you're trying to see whether the disease is cured. You're not worried about the target in the initial stages. Now, once if you feel that the disease is getting cured, then you go on to develop which is the target is this molecule is affecting, and then you go for target identification, and then there is a lead optimization, and then it moves on to the same route as earlier. Now, drug repurposing is another way of drug development where you already have a drug in the market, but which can be repurposed for something else. For example, if you take aspirin. Aspirin was initially used as an analgesic, but then later on now it is repurposed as a platelet aggregation inhibitor. So now aspirin at a lower dose is given for people who have a high level of coagulation and to prevent coagulation. So this is known as repurposing. And then target identification. So this is the same target identification for repurposing a drug using phenotypical staining. So these are different methods of screening and development which are used in the development of a drug. Now, this is just to make you understand that what is target based. So target based is there is only one target, which is you're only trying to understand if my molecule will inhibit this target. But then in a phenotypic, you have so many pathways which are involved in the disease. So you're trying to understand which molecule is going to act best on all these targets, and then give you a relief from the disease. Now, high throughput screening is one of the very, very important aspect of drug discovery, which now has like earlier when discovery started, probably we were using test tubes to do the enzyme assays, and then you use spectrophotometer one by one. You are trying to take the reading. Then it came 96 wells. Now you have 384, 1536, 34, like 3,456 samples could be done at the same time. So this is high throughput screening. So you already have the everything is miniaturized, and then you are able to identify that molecule which is having the maximum inhibition or maximum activity, and then from there you go on to develop which can be expanded further. Now, then as I told you earlier, this is the hit is the one which is giving you the activity. Then from there you develop the lead molecule, and then the lead molecule needs to be optimized. What is optimization? When some structural modification which can increase the activity and which can have proper pharmacokinetic activities, and then which can also have lower toxicity. So these are the aspects which are involved in lead optimization. So once you have a lead which is optimized, then we move on to the preclinical research, where the most important aspect of preclinical research is to see whether the efficacy is what you see in the in vitro screening system is translated into an animal model. And then the absorption, distribution, metabolism, and excretion. These are the different aspects of the drug which needs to be studied. And then the side effects or the adverse effects, which are very important. And then how is it compared to other similar drugs of the same category? So these are some of the aspects which are taken care of in the preclinical research. So for and then we move on to the clinical, where you have the phase 1, the phase 2, and the phase 3. The phase 1 being only the side effects or the mainly to study the safety. Then move on to phase 2, where it is around 500 people or a limited number of individuals, where it is compared with a placebo or a dummy drug. And then we move on to the phase 3, where it is done at multiple sites and compared to other treatments and placebo. And then it moves on to the post-marketing survey, which is a phase 4. Now, after phase 3, you can go for an FDA review, and then FDA can approve your drug for human therapy. And then like when the drug is in the market, there is a continuous post-market market monitoring. And this is very important because initially the drug is being developed only in a small population. So when it is given worldwide in the global population with different ethnicity, different way of reacting to the drug, so a lot of drugs can fall at this post-market scenario. So one of the recent example probably is ranitidine, which was recently withdrawn because it offers side effects. So there are several reasons for drug failure, and you start with enormous number of molecules, maybe thousands, which you probably take only a few of them to the preclinical stage, and only probably like 250, if they are going to preclinical, only 5 of them will probably qualify for a clinical phase. And then from there, you will probably get only one which is safe, which is a drug which is approved in the market. Now, the time taken is very long. Like you start maybe more than 10 years, 12, 20 years it takes for the drug to come into the market. And the cost involved, of course, is really huge for any pharmaceutical company. So having this background on what is drug discovery, we move on to see how biotechnology has helped in speeding up this drug discovery process. So biotechnology is involved probably in all the aspects of drug discovery, from raw material analysis to the lead discovery, the efficacy. And the most important aspect of biotechnology has been in the discovery phase, where you have developed new methods for high throughput screening, and different models have been like we just listened to the earlier talk where we were the knockout and knock-in animal models were being discussed. So these knockout animals are very, very important when it comes to drug discovery. So some of the disease models, you need to have a knockout model to understand whether the preclinical model, at the preclinical stage, whether the drug is working. So again, going on to to little more details, the automated techniques, the genomics, by all these, like I've just put them in one a slide to show that so many aspects of biotechnology which are involved in drug discovery. Now, apart from this, the recent ones are the AI, or the artificial intelligence, which is involved in, which is being used in drug discovery. Then you have organoid system, which helps immensely in understanding the in vitro efficacy of a drug. The organ in chips, biosensors, the microfluidics, which has tremendously helped in high throughput screening. So, tell me if I'm going too fast. So I'll move on to the natural therapeutic agents. So what are these natural therapeutic agents? Now, we've been talking about drug discovery as a chemical moiety. So most of the drugs which are present in the, like, which are being in the discovery stage, are synthetic molecules which are synthesized in the chemistry lab based on different. They do get a lot of clue from natural chemistry, but then they're all synthetic molecules. Now we are talking about natural therapeutic agents, which are the most dominant the source of natural therapeutics is the plants or the herbs, which have enormous chemical and structural diversity, and then they have been known for years together for treating different diseases. So when they are identified as a lead, they can become candidates for drug development. In fact, more than 60% of the drugs which are in the market are derived from natural sources. So, for example, if you take morphine, morphine is one of the oldest analgesic which is there in the market, which was taken from a plant called Papaver somniferum. So, and then you have paclitaxel, which is from Taxus species, which is now a very well-known anti-cancer drug. So many of these products like are also FDA approved. For example, you have for cardiovascular system, you have omega-3 ethyl esters, analgesic, which is, these are all, I'm just given you a few examples which are taken, the source has come from plants, and then they have been structurally modified a little so that they follow the rules of drug discovery. That is, they have a good absorption kinetics, good pharmacokinetics, and lower toxicity. So all these aspects are synthetically, they are just modified from the natural source, and that is approved by the FDA for drug. Now, why are they so attractive, the natural products? So they have always played a key role in drug discovery. Now, especially for cancer, infectious diseases, and diabetes also, they have, the natural products are, they have a huge diversity in the structure, and then they are structurally optimized by evolution to carry out a particular function, because through natural evolution they have been like they have been optimized for the structures have been optimized. And then traditional medicine, which gives you an idea about their role in a particular disease. And then it also gives you an idea about efficacy and safety. So that is that these are few of the reasons why natural products are so attractive for drug discovery. Now, there are definitely some difficulties, several drawbacks, which have led to main the leading pharmaceutical companies to reduce the natural product based drug discovery. Now, what are the main problems? They involve a library of extracts from natural sources, which may not be compatible with traditional target based assays. So for example, most of these natural products have a huge problem in solubility. Now, you need to use DMSO or some solvent to dissolve these compounds. Now, once you dissolve this compound and when you put them in a buffer, like most of the assays, high throughput assays are done in buffers. Now, when you put them in a buffer, they precipitate out. So how do you solve this problem? So this has been a one of the limitations for using natural products in high throughput typical target based assays. So and then you and then identifying, so the plant has so many secondary metabolites and so many products, so identifying one particular compound, a purified compound, which can be taken up for further research, that could be another very challenging aspect, because purification can be very difficult. Every plant does not produce one particular product in large amount, so the amount that is present may be so less that it may not be economically viable to produce them in a large quantity. And then getting sufficient biological material to isolate and characterize these natural products is again next biggest problem that we have, because like the source is limiting. Unless you have your own farm where you're continuously producing these plants, the plant, like for the synthesis of, so for the drug molecule to in such a large quantity may be a biggest challenge. Then intellectual property, another important aspect of drug discovery, because any new product which is developed by a pharmaceutical industry, it is generally patented, and then the intellectual property may be a problem when it comes to natural products, or if it's a well known traditionally used product for a particular disease, then IP rights may not be possible to get. So the even though you are spending so much money on drug discovery, you may not be ultimately able to reap the benefit because you cannot patent the molecule. So a few example from what we've been doing so far. So I will talk about one or two examples where we have tried to identify these problems, challenges which I talked about, and how we overcame those problems. So one example is, all of you have heard about the turmeric or the curcumin from turmeric, which we use every day. Now, the turmeric generally gets metabolized into tetrahydro. The curcumin is one of the major bioactive molecule which is present in turmeric rhizome. It gets metabolized into tetrahydrocurcumin in the intestine, and which is biologically active. So a simple hydrogenation of the natural product will give you the turmeric, which is white in color, the tetrahydro part. Now, tetrahydrocurcuminoids it's colorless, and it's an active metabolite and has the advantage of higher bioavailability than the curcumin, which is yellow in color. Now, we have this product in as one of our major selling product, that like a simple chemical reaction could give you a product which has better bioavailability, and which does not have the color, which can be a problem when using in different formulations. So this is one of the simplest way our company came across a product which can be used in different formulations. Now, the second example, again, I'm talking about the same plant. Apart from the curcumin, which is very well known, there is one more product called calibin, which is present in the same plant. But it is present in such a minute quantity, that 0.01%, that it cannot be isolated from the plant. So what is the way to produce it? So we found that there are some endophytic fungi which produce this monooxygenase, which can biotransform curcumin to calibin, and it could also be chemically synthesized for commercial application. So a product which is present in the natural plant, which is in a very, very low concentration, could not be actually was not economically viable, was made economically viable by either chemically synthesizing or having a different approach to synthesize it. And then now this is one of the product which is in the market. Now I'll go on to elaborate on one aspect of novel drug discovery, where the biotechnological tools were used to the maximum extent. Now, this is a drug for there is a development of a novel drug for obesity. Obesity, as you know, is one of the major problems in the society right now, and there are no drugs which are available to reduce obesity. Of course, I also believe that it cannot you cannot pop a pill and say I will reduce weight. It is just not possible unless you have other control in your lifestyle changes are taken along with the drug. Now, morbidly obese people, the drugs can be definitely be one of the choice. Now, just to let you know about the biotechnology in this drug discovery process, leptin is an hormone which is produced by the adipocytes or the fat tissue. Now, this leptin goes to the brain and tells your system that there is enough energy, so stop eating. So this is one of the hormone which tells you to stop eating when you're full. Now, what happens in obesity, the condition of obesity is something called as leptin resistance develops. So when even though you have a lot of leptin, it is not able to convey the message to the to your brain or the hypothalamus to saying that there is enough energy balance available in your system. Now, increased endoplasmic reticulum stress was supposed to be the central role which for the development of leptin resistance. Now, what is ER stress? So, ER stress is a condition when your cellular demand for protein synthesis, the demand for is very high, and the ER is not able to support the synthesis by producing so much protein. So what is happening here is there is excess of proteins which are being produced, but they are not getting unfolded, or they are protein folding is not happening properly because ER is not able to match with the rate of protein synthesis. So there is a lot of malfolded proteins accumulating in the endoplasmic reticulum, which is causing stress. And this particular ER stress is known to have a role in all the metabolic diseases. So, in this case, what they found that increased endoplasmic reticular stress in the brain plays a central role in the development of leptin resistance and consequently obesity. So here, so the aim and approach, this is not our work, but I'm talking about a work which was where the aim of the study, drug discovery, was to identify a small molecule that could increase leptin sensitivity. So they used a connectivity map for this case. So connectivity map is a whole genome gene expression profile derived from human cell lines, which were treated with thousands of small molecules. Now, the software can compare two sets of genes which are upregulated and downregulated and for a specific condition. So, in this case, what was done was the gene expression signatures were created using microarray data obtained from following various by giving some interventions to relieve ER stress. So, that is one aspect. Now here, the liver and hypothalamus of obese and lean mice were treated with this 4-phenylbutyrate, which is an ER stress reducer. Now, the microarray data which is obtained from this animals, the obese and lean mice, were compared with the CMAP data, which is already been which is there in the database, which has the molecular signatures for for different small molecules. So when they compared this, they identified two different small molecules which are cholesterol and beta-ferrin A. So they found that both these could suppress food intake and increase energy expenditure. So based on this published data, so what we did was we understand that ER stress is an important aspect for obesity. So how can we use this ER stress phenomena to understand whether we have any of the natural product which are targeting this ER stress. So we went on to, we have a molecule called garcinol, which is derived from a plant called the fruit called kokum. Kokum is it's a fruit which is present in the Konkan area, the Goa that belt, which is used normally in as a alternative for tamarind maybe. So it's used very frequently in these areas.
Dr. Lakshmi, sorry to interrupt you. Can you
Yeah.
please try to end?
Yeah, I understand. I'm just taking another few minutes.
Sure.
So, so what we did was we did some in vitro screening of this ER. We only focused on ER stress and looked at garcinol to reduce the protein aggregation in the liver cells. And also we looked at some of these proteins, spliced XPP, and then the like GRP78. So we looked at it in both liver cells and adipocytes. So based on this, we thought garcinol could be an ER stress reliever. Now, when we took it for this is another phenotypic screening that we do in vitro. So we take the pre-adipocytes and convert them into adipocytes by different induction process. And then we found that this garcinol at different concentration could actually prevent the lipid accumulation in adipocytes. And this, when it was taken to preclinical, in an animal model which was given high-fat diet, now we find that garcinol could definitely reduce the weight accumulation, high-fat induced weight in animal models. So we went on to characterize different proteins, different cell pathways, and then we published this in Journal of Functional Foods. So what I'm trying to say is, based on different biotechnological processes, you can actually come up with different strategies for screening and discovery using natural products, or of course the same thing can be used for synthetic products also. So the use of advances in genomics, and this will help in searching for novel products from nature and applying new screening technologies would be the way forward for drug discovery. So thank you for your attention, and if you have any questions,
Thank you so much, Dr. Lakshmi, for this excellent presentation on the drug discovery process. I'm sure the students would have learned a lot. Any of you have any questions? Please don't hesitate to ask.
Can I ask you one question?
Yeah, sure, definitely.
Yeah, we have this drug discovery. It was a wonderful talk, and this drug discovery program, from in vitro to the human clinical trials. So is there any particular rule in India or in other countries? Like we do in vitro assays, then we go on to the in vivo assay where we limit our experiments with the mouse models. Is there any particular rule that we need to do in large animals, or will it be better as per the rules of our government and ICMR or drug discovery unit?
So actually, large animal studies are not really much approved nowadays, because unless it is because there's a lot of the PETA rules which are now coming into play, and they don't allow large animals to be used for drug discovery. So now there are a lot of that's where the biotechnology also plays a lot of rules, where the toxicity part is also taken care of by a lot of structure activity relationships. So there are tools available now, softwares available, which tell you a lot about the toxicity of the drug in the by in silico and screening. And then the FDA also has a enormous set of rules where you need to do the preclinical toxicity. So the OECD guidelines, and then as per the guidelines, there are several battery of tests which have to be done before you can even take it for the first phase of clinical trial. So you don't really need to go into the large animal model, but you need to have all the safety records set before you can take it for the first phase of human trial.
Thank you. Thank you very much.
Yeah. So one just comment in addition to that, what Dr. Lakshmi said. Now this lot of organoid
Yeah.
cultures are also picking up. I think lot of people are doing
Yes.
organoid models for testing of the various drugs in the preclinical level.
Yeah.
That is another exciting area that I think is also picking up. Because of the restriction in large animal testing,
Yes.
that is one kind of area that people are now utilizing. Any other questions?
Yeah, to add
Maybe.
to it, there are like now stem cells which are being used for testing, and as you said, organoids, and then reduce animals is what is the mantra now, because
Right.
yeah, reduce the testing in animals.
Yeah. So if no other questions, I think we will kind of thank Dr. Lakshmi for the wonderful talk, and then we will close the session here and take the lunch break.